what we have in the test tube is the competent E. coli cells with this pGLO DNA. And I want to explain what this plasmid is. And so this is roughly a map of this plasmid. And that's what the P stands for. But what is a plasmid? Right, it's extra chromosomal DNA. How does it compare size-wise to a chromosome? It's tiny, right? It's a lot smaller. And that makes it convenient for such procedures like transformation and other things. These do occur naturally as well. So bacteria do have plasmids and they make use of them. And it's when we go over lateral transfer next week, we'll see even more about why these are useful for bacteria. And so the little P stands for plasmid. We call this the pGLO plasmid, and the GLO is sort of the brand name. Now, it gets that as a brand name in part from one of the genes that's contained on this plasmid. And I've already given a little label or abbreviation for it, and I'm calling it GFP. Does anybody know what GFP stands for? Yeah, Mitchell? Yeah, the green fluorescent protein, right? Do you know where it comes from? Yeah, right. It's originally a jellyfish gene. And in 2 weeks, we're going to have a lecture on recombinant DNA and genetic engineering. So we'll see how it's possible to get this jellyfish gene into this plasmid and then into bacteria. But for now, we'll just say that there's this plasmid with a jellyfish gene, green fluorescent protein. And maybe many of you have seen exhibits or images or real jellyfish up close, and a lot of them do glow. They have a fluorescence to them. And that's where this gene comes from, a fluorescent jellyfish. And so if this procedure works correctly, we will have transformed the E. coli so that they have this jellyfish green fluorescent protein and will glow green. And so again, if it works properly on our plates next time, our nutrient agar plates, we'll have these colonies that glow green. Now the truth is we have to shine UV light on them so they absorb the ultraviolet light and then fluoresce green as a result of that. But it'll still be really cool. We'll see these glowing colonies. Now, I'm going to say more about GFP and why we do this in a bit, but there's one more gene on this plasmid. And that gene often has the name AMPR. Any ideas what that is? Yes, ampicillin resistance. So someone else, what's ampicillin? Yes, it's a penicillin-type antibiotic, right? It targets the cell wall, thank you. And so if the bacteria have an ampicillin resistance gene, what do you think is probably happening? Like, what do you think this gene is making? Yeah, probably a beta-lactamase enzyme, right? Which will then do what to the ampicillin? Yeah, break open that beta-lactam ring, inactivating the antibiotic. Great. And so, now, that might sound strange. What the heck are we doing, right? We're putting an ampicillin resistance gene into the bacteria. Why would we do such a thing? Mitchell, yeah.
Markers that can grow out of control what you collect.
Right, yeah. So we use it as a selectable marker. What we're going to do in this experiment is we're going to plate our bacteria on nutrient agar plates that have ampicillin in them. Now, the reason we do this is because the procedure is very inefficient. So we're taking these, hundreds of thousands of E. coli cells, mixing them with the DNA, and probably only a handful of the bacteria will actually get transformed. And so we're talking much less than 1% of our cells. And so if we were to plate them on nutrient agar, everything would grow, and the glowing green colonies would be lost in this huge lawn or sea of bacteria. And so what we do is we plate the cells on nutrient agar with ampicillin in it, and the ampicillin is going to kill 99.99 something percent of the bacteria. The only ones that survive have pGLO in it, because again, they're resistant to the ampicillin. And so we get these nice visible colonies that also have GFP in them, and so they're glowing green. And so we called it a selectable marker, right? So we grow them on ampicillin plates, and we're selecting for those that have ampicillin resistance, right? They're the only ones that are going to survive and grow. Questions about the plasma? Okay, so this is, yeah, go ahead, Alexei.
So there are only 2 genes in the plasma?
There might be others, I'm not sure what they are, but there could be others as well. Yeah, but these are the only 2 we're going to observe in it. Now, this is a nice demonstration of the principle of transformation and of the technique of it in a laboratory. And we get the cool glowing green colonies. But this has a lot of applications. So, for instance, in a lot of, in the pharmaceutical industry, they might, instead of GFP, have the human insulin gene inserted into this plasmid. Who cares about insulin? Diabetics, right? And so about 30 years ago, a little more maybe, at Genentech, a local biotech company, they developed a technique where they took the human insulin gene and got microorganisms to produce the insulin for them. They transformed those microbes with the plasmid like this. And when you do that, you can then grow these huge industrial-sized vats of the microbes that are all have the human insulin gene in there, and they make the insulin. And so then you take those cells, you can harvest and purify the insulin away from them, and now you have a very cheap and easy to produce pharmaceutical. And so nowadays, that's how almost all insulin is produced. Microorganisms make it for us in this way and following a transformation procedure. Now, that's a cheap and easy way to make it. 30 years ago, how did we get our insulin? Anyone know? From animals. Does anyone know which animal? Generally from pigs. Pig insulin works pretty well in humans, but of course you have to raise pigs, you have to slaughter pigs, you have to get the pancreas and purify insulin from there. And so that's a lot of work. Doing it this way in bacteria or other microorganisms is very easy by comparison. You can do it on an industrial scale for, relatively low cost. And so that's how we do this nowadays with insulin. We do it with a lot of other pharmaceuticals. We do it with growth hormones. We do it with clotting factors. Who needs clotting factors? Hemophiliacs, right? And so we get it that way. And we do this with dozens of other pharmaceuticals as well. And so, again, we're doing a nice demonstration of the procedure, but this just has a lot of application.